Hello everyone and welcome. I'm your host for today's webinar, Jessica Tu from the ProBio Marketing Department. On behalf of ProBio, I'd like to extend my sincere appreciation for your participation in today's session. ProBio is a global leader in enabling biotech and pharmaceutical companies to advance the development and manufacturing of next-generation biologics and advanced therapies. As a fully integrated end-to-end contract development and manufacturing organization, ProBio supports its partners in optimizing drug development, accelerating time to market, and provides comprehensive life cycle support. Today's webinar will feature 3 presentations. Each talk will include a 30-minute presentation, followed by a 15-minute Q&A session. You are encouraged to submit your questions at any point during the webinar using the chat or Q&A panel located in the menu on the right. Now, I'm delighted to introduce today's first speaker, Mr. Luffy Chen, the head of Plasmid PD and Production Department of ProBio. Luffy has 5 years of experience in Plasmid CMC, from preclinical CMC studies to clinical trials. Luffy obtained his Master of Science degree at Huazhong Agricultural University. During his 8 years industrial practice, he devoted to the development of new technology platform, meeting the fast-growing market and customer demand, and helping customers improve production quality and efficiency. During today's webinar, Luffy will be discussing plasmid applications and production challenges in cell and gene therapy. Now, I'm glad to hand it over to Luffy.
Thank you, Jessica.
You're welcome. You can share your screen.
Okay. Can you see my screen and hear me clearly?
Not yet. Could you share it again?
Sure.
I can hear you, yeah, but I can't see your screen yet.
How about now?
Yeah, sure, you can play it. Good.
Very warm welcome to this webinar. I'm really delighted and excited to talk about some plasmid DNA applications and manufacturing challenges in gene and cell therapy. There are 3 sections in my presentation: plasmid DNA application in gene and cell therapy, plasmid DNA manufacturing challenge and strategies. Finally, there will be some case study. Firstly, I will talk about what plasmid is and its role in gene and cell therapy. A plasmid is a small DNA molecule found in bacteria. It is separated from genomic DNA and can replicate independently. They typically have a small number of genes, antibiotic-resistant gene, origin of replication, and the gene of interest. On the one hand, it serves as a raw material for gene and cell therapy, usually as viral vectors to package like lentivirus vectors, adeno-associated virus, vitro virus, and deliver functional genes to patients in vitro, ex vivo, or in vivo. On the other hand, it can also work as a in vitro transcript template for mRNA therapeutics, like mRNA vaccines, mRNA-based therapeutics. Here, we make a brief summary about plasmid type, features, applications, and some clinical cases. First one, supercoiled plasmid. It is widely used in GCT industry. It usually shows high yield and the manufacturing process is scalable and robust. It serves as a raw material for gene and cell therapy, or mRNA therapeutics, or DNA drug or vaccine. For example, Parikti from Legend and J&J, which is one of the most successful party therapeutics. The supercoiled plasmid encodes genes for LVV lentivirus package and T-cell engineering. Another gene therapy clinical case is Zolgensma from Novartis. It is AAV vector-based medicine for treating SMA. There are also some other types of plasmid used in GCT. Mini circle, also called nanoplasmid or gene circle. It is marker-free and smaller than traditional plasmid, so it has reduced transfection toxicity, and which is a good vector for DNA vaccine or DNA drugs. Linearized plasmid, this shows high in vitro transcript efficiency, is an ideal template for mRNA therapeutics or vaccines. And also linear closed DNA only consists of gene of interest. There is no bacterial sequence. So it is much safe and is usually work as a non-viral donor for cell therapy or gene editing. Single-strand DNA, ssDNA, it can also work as a donor plasmid for gene editing, but since it is single-strand, not stable as double-strand DNA, so far there is few clinical trials. For different application plasmid, we have several manufacturing strategies to meet our customers' needs. For example, linearized plasmid for IVT mRNA use. We have process 1.0, ProPlus process, and ProSign process. Process 1.0 is the classical one. Firstly, make a supercoiled plasmid with multiple chromatography steps and QC release. Then produce linearized plasmid. Both supercoiled plasmid and linearized plasmid are deliverables, and the supercoiled plasmid can stored for many years as a raw material for mRNA. ProPlus process, we reduced one or two steps of chromatography, and there is no QC release for supercoiled plasmid. So the production timeline and cost can be reduced a lot, around 50% compared to process 1.0. Here we show an example for ProPlus process performance. You can see there is compared to process 1.0, Plus process consists of less steps, so less production timeline, higher yield, higher recovery rate. So for GMP production, usually the simple process is good. Personalized mRNA vaccine provide a new directions for precision tumor treatment with existing clinical data and a large number of clinical trials. Personalized mRNA vaccine have potential as a adjuvant therapy for solid tumor, but the PCV mRNA production timeline is the bottleneck. it is required that PCV mRNA must be delivered in 3 weeks from template plasmid synthesis to mRNA LNP delivered. The traditional plasmid manufacturing process based on bacteria could not meet this requirement. So we developed cell-free plasmid manufacturing process. We call it ProSign process to produce linearized plasmid for PCV mRNA. Our ProSign process utilizes rolling circle amplification, and there's no cell bunking, no fermentation steps, which can obviously cut plasmid production timeline from several weeks to several days. Moreover, the scale and the small devices required in ProSign process only occupied a small area in our GMP facility, and which is easy and flexible to operate. This will help a lot to accelerate DNA to mRNA manufacturing. Here we showed an example for our ProSign linearized plasmid performance from circular plasmid to mRNA LNP. The red one is the cell-free, linearized plasmid DNA made by ProSign process, also cell-free process. The black one is the linearized DNA or mRNA made by E. coli, traditional bacterial system, and we can see the ProSign linearized plasmid quality and its mRNA is comparable with E. coli, especially for the critical quality attributes, the poly purity and integrity. Cross-contamination is one of the biggest challenges in plasmid manufacturing. The root cause for cross-contamination ranges from GMP facility to technical issues. We have developed a control strategy to prevent cross-contamination from environment, procedure, personnel, facility, and equipment. For example, our regular environment monitoring and the validated clearance procedure, cleaning qualification, validation for residual plasmid by qPCR or NGS test. And we have a strict gowning system for personnel to enter our clean rooms in GMP facility, and also APS is required for final filling and finish operation. Considering the different applications of plasmid in GCT, we suggest to choose different grade of plasmid for different phase of clinical studies, like pre-CMC or pre-CMC grade plasmid for pre-clinical study, GMPro grade plasmid for phase 1 and phase 2, GMP grade for phase 3 and commercial use. Let me now give you some examples about the plasmid manufacturing challenges we made. First one is in a vaccine plasmid. The key pain point is the plasmid yield is not high as expected. So we did the process development, like for the temperature shift fermentation for upstream process, the feeding medium component, and 2 steps of chromatography instead of traditional 3 steps of chromatography for upstream process. After process optimization, the fermentation yield increased around 3 times from less than 1 gram per liter to over 3 gram per liter. And the total recovery rate increased 3 times from around 14% to 41%. If this DNA vaccine, the dose is around 3 milligram per patient for a 100-liter fermentation, we can finally obtain almost 40,000 doses, which increased around 10 times. The second case is Plasmid used for self-amplifying mRNA, saRNA. In our PD batch, we found unexpected hence after BSPQ1 linearization, and the purity of linearized plasmid is only 75% by IC-HPLC test. We did a complete investigation of this issue from our production process to critical materials, and finally we found there are several BSPQ1 star activity site in the plasmid. The star activity size is only 1 BP difference than the BSPQ1 recognition site. So we proposed 2 plans for our customer. Plan A, optimize plasmid sequence to avoid BSPQ1 star activity site. Plan B, optimize the BSPQ1 digestion process to lower star activity. Since our customer is unwilling to make any change in the plasmid sequence, so we proceed this project with Plan B. After rounds of process optimization, we eventually improved the linearized plasmid purity to 95%, and the BSPQ1 star activity is under control. Here there's some production data about this project, the scale-up batch, the chromatography map, AG gel, and the IC-HPLC of final linearized plasmid 95%, and some critical quality attributes for the final linearized plasmid. HCD, the total protein residue by micro-BCA. Also, we list some tips here that may help to avoid star activity in the digestion process. The third case is about a plasmid used for in vitro transcript mRNA. Our customer said that our deliverable linearized plasmid is not pure and shows smaller bands on AG analysis. Here, the red arrow is the smaller bands. The yellow one is our target linearized plasmid. The smaller one affect customers following IVT mRNA purity. Our scientists looked up some papers and did some small scale of experiments, and finally we found that it is not the purity issue of our deliverable linearized plasmid. It is another structure transition. When the B form DNA, our normal DNA, linearized DNA, is diluted in water at a concentration of lower than 0.1 milligram per milliliter, there might be the structure transition from B form DNA to A form DNA. So we recommend the storage buffer for linearized plasmid water for injection at a higher concentration, like more than 0.1 milligram per milliliter, or a solvent buffer with a liter, like 10 millimole trace. And the B form linearized plasmid is a higher quality plasmid, which has a higher IVT mRNA purity. Yeah. So that's all for my presentation about the plasmid manufacturing challenges and strategies. I will stop here, and feel free to let us know if you have any question about and comments about our presentation.
Thanks for Luffy's excellent presentation. We will now open the floor for questions. Please type your question in the chat or Q&A panel. Additionally, we also invite you to follow our LinkedIn account, ProBio, and the Korean attendees can also scan the QR code on the screen to follow our Kakao channel, where we will regularly share our online resources and updates there. Hi, Luffy. Here is a questions. What are the steps taken to improve the plasmid DNA yield as shown in the talk? Let me copy and send to the chat box so everyone can see it clearly. Could you please answer this question?
Sure. Usually, it is the upstream process, like bacterial fermentation. Our upstream process scientists usually to develop firstly temperature shift to highly increase the fermentation yield. Also, the feeding media component, the different carbon and nitrogen ratio will also affect the plasmid yield in bacteria. Yeah, usually these two key steps to increase fermentation yield for plasmid in bacteria. First, the temperature shift and the feeding media component.
Thanks for your answer. Hope this answered the question from the questioner. And here is the second question. What's the advantage of protein DNA process compared to E. coli manufacturing?
Thank you. It's a good question. Our protein process utilize rolling circle amplification. There is no cell bunking, no fermentation, no supercoiled plasmid process. So the production timeline can reduced from several weeks compared to E. coli process. to several days. That's a significant improvement, especially for the plasmid used in personalized mRNA cancer vaccine.
Thanks for sharing. And the following question is, you showed a slide with different requirements for preclinical, phase 1, etc. Where did you get this info from?
It is prepared. It is made by our internal team, our QA, our RA team. They make this table according to the admin, according to the NMPA, FDA, or EMA guidelines. So yeah.
Yeah, thank you for the answer. So this actually made by our overall teams together, and then gather this information and share with everyone about our customized solutions. And here is another question in the Q&A panel. Besides the quality controls, is there any performance difference among the various grade of plasmid?
Sorry, viral grade plasmid.
Yeah, I copy and send the chat box so you can check there. Besides the quality control, is there any performance difference among the various grades of plasmids?
Let me see. Usually for different grade of plasmid, the manufacturing process is the technology is the same. We based like coli or cell-free system to make plasmid. Just maybe the facility is different. Like pre-CMC grade plasmid is manufactured in our CNC area. GMPro grade plasmid is made in our GMPro U facility, and the GMP grade plasmid is produced in our GMP facility. Just the technology is the same. The quality standard is the same. The quality system, the background, the equipment may be different.
Thanks for your clarification. And next question is, what modified mRNA people usually use for cell therapy application? Could you answer this question?
It's a good question. Yeah, but I'm a scientist for plasmid process development. Yeah, I have a lot of experience in supercoiled plasmid or linearized plasmid production, but I'm not quite familiar with mRNA. Yeah, so I'm not quite sure what modified mRNA the other customer should use for cell therapy. Yeah,
No
maybe
problem.
I can check with our internal mRNA department and get back to you.
Yeah. We'll reply the email for you to answer this question after this webinar. Thanks for your question. And here comes another question. Is there any requirement of running supercoiled later along with in-process pDNA samples?
Supercoiled runner. Jessica, could you repeat this question? Oh, I can see. Is there any requirement of running supercoiled ladder? No, this is our internal SOP from our AD or QC team. Since we make supercoiled plasmid, also linearized plasmid, usually when we run the AG gel, we will load supercoiled plasmid and linear ladder as a control. So we can see how the supercoiled plasmid is performed compared to supercoiled ladder, and how linearized plasmid performed compared to like KB DNA ladder. It's not a guideline or regular. It's just our internal procedure, not what we must do.
Thank you for your answer. Here are all the questions I received by now. And thank you all for your engagement and insightful questions. If you have any further questions, feel free to leave a message to us through our email. Thank you, Luffy.